Today we are going to learn about gluconeogenesis. What is gluconeogenesis? Gluconeogenesis is the synthesis of glucose from the non-carbohydrate compounds like lactate, pyruvate, glucogenic amino acids, propionate, and glycerol. The gluconeogenesis mainly occurs in the cytosol and some part occurs in the mitochondria. Around 1 kg of glucose is synthesized every day in the liver, and some part of this gluconeogenesis is synthesized in the kidney, and mainly the gluconeogenesis takes place in the liver. Why it is important, why gluconeogenesis is important? The mainly brain and that is the central nervous system, erythrocytes, testis, kidney, medulla are dependent on the glucose for continuous supply of energy, and a human brain alone requires around 120 gram of glucose per day, and around 160 gram needed for the entire body. That's why for the supply of energy to the brain, central nervous system, this gluconeogenesis is very important. And in fasting level also, the gluconeogenesis meets our body's basal energy requirements, and therefore for survival of human beings and some other animals, this gluconeogenesis is very important. There are total three reactions in the three steps in the gluconeogenesis. As I already told you, the gluconeogenesis is the reversal of glycolysis. It is also called as a reversal of glycolysis, but not complete reversal of glycolysis, because already in the glycolysis we have discussed that the total there are 10 steps in the glycolysis, and out of that the 3 steps are irreversible. So for those 3 steps which are irreversible, the different enzymes are used in the gluconeogenesis. So those 3 irreversible steps are important, and that we are going to discuss in the gluconeogenesis. So the 7 reactions for the glycolysis are same for both glycolysis and gluconeogenesis, but the 3 irreversible steps which are there in the glycolysis are catalyzed by using different enzymes in the gluconeogenesis. So mainly those 3 enzymes for glycolysis were hexokinase, phosphofructokinase, and pyruvate kinase. So for these 3 enzymes here in the gluconeogenesis, we will use the different enzymes. So the first step of the gluconeogenesis is the conversion of pyruvate to phosphoenol pyruvate. So starting from the end of the glycolysis, the end product of glycolysis is the pyruvate. So here in the gluconeogenesis, we are going to start from the end, okay, because we want to synthesize the glucose. So first step will be the end step of the glycolysis. For gluconeogenesis, the first step will be the end step of glycolysis, and here the first step will be conversion of pyruvate to phosphoenol pyruvate. In glycolysis, for this step, we have used the enzyme pyruvate kinase. But as this step is irreversible in the glycolysis, here we are going to use the different enzymes. So in the gluconeogenesis, we are going to use 2 different enzymes instead of the pyruvate kinase. The first enzyme is the pyruvate carboxylase, and the second enzyme is the phosphoenol pyruvate carboxykinase. So instead of pyruvate kinase, here we are going to use these 2 different enzymes. Now, some part of this step occurs in the mitochondrial matrix, and some part occurs in the cytosol. So mainly here these 2 enzymes are important in this step, and the second step will be the second irreversible step will be the fructose 1,6 biphosphate to fructose 6 phosphate. For here this step also we are going to use the different enzyme, that is fructose 1,6 biphosphatase. In the third step, the conversion of glucose 6 phosphate to glucose. For this step also we are going to use the different enzyme, that is the glucose 6 phosphatase. And then we will get finally the synthesis of glucose in the gluconeogenesis. If we can see one by one the steps of the gluconeogenesis, the first step, see if you can see in this figure, the first step will be the conversion of the pyruvate to phosphoenol pyruvate. So here they have given that pyruvate as the enzymes which are required, this is a irreversible step in the glycolysis, because pyruvate kinase enzyme we have to replace here. So we can't convert directly pyruvate to phosphoenol pyruvate. So for that we have to use 2 different enzymes, that is the pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Now what will happen here at this step, the pyruvate first enters into the mitochondria, because the enzyme which will convert pyruvate to phosphoenol pyruvate are not present in the cytosol. They are present in the mitochondria, the enzyme pyruvate carboxylase. So for this enzyme, pyruvate enters into the mitochondria. Then pyruvate first convert to oxaloacetate by the use of enzyme pyruvate carboxylase. But oxaloacetate can't cross the mitochondrial membrane. And therefore, this oxaloacetate is converted again to malate by the use of enzyme malate dehydrogenase, and then malate cross this mitochondrial membrane. Okay, after that, then malate enters into the cytosol, and then again malate converted to the oxaloacetate by the use of same enzyme malate dehydrogenase, and then this oxaloacetate then converted to the phosphoenol pyruvate by the use of enzyme phosphoenol pyruvate carboxykinase. So simply in the glycolysis, we have used simple one enzyme, phosphoenol pyruvate to pyruvate for conversion. But as this is irreversible step, in the gluconeogenesis, we have to use these two enzymes, pyruvate, that is the pyruvate carboxylase, and phosphoenol pyruvate carboxykinase for the conversion of pyruvate to phosphoenol pyruvate. So these are the two different enzymes we have used here, and this is the first step of the gluconeogenesis. What we have covered in this step, as pyruvate can't cross the mitochondrial membrane, or pyruvate we have to convert to the phosphoenol pyruvate. So then this pyruvate enters into the mitochondria first. Then this enzyme is not present in the cytosol, pyruvate carboxylase. So pyruvate enters into the mitochondria, and then pyruvate first convert to oxaloacetate. Now as oxaloacetate can't cross this mitochondrial membrane, it first convert to the malate. Then malate enters into the cytosol, malate again converted to the oxaloacetate, and then oxaloacetate finally converted into the phosphoenol pyruvate, and this is enzyme used here is the phosphoenol pyruvate carboxykinase. So this is the first step. At this step, we get conversion of pyruvate to phosphoenol pyruvate. Then after that, the reversible steps are there. See, phosphoenol pyruvate to, we will get formation of 2-phosphoglycerate. Reversible, all steps are reversible, so same enzymes we will use here. Okay, then 3-phosphoglycerate, then 1,3-biphosphoglycerate, then glyceraldehyde 3-phosphate, then fructose 1,6-biphosphate. Till here all the reversible steps are there, so they will occur as same in the glycolysis reversibly. So till we get the formation of fructose 1,6-biphosphate. Then the second irreversible step here is the fructose 1,6-biphosphate to fructose 6-phosphate. At this step in the glycolysis, we have used the enzyme phosphofructokinase. Now we have to replace this enzyme. So in the gluconeogenesis, we will use the enzyme fructose 1,6-biphosphatase. So we will get the formation of fructose 6-phosphate here. Then again, this is reversible step, fructose 6-phosphate converted to the glucose 6-phosphate. Then again, this is a irreversible step. Here in the glycolysis, we have used the enzyme hexokinase or glucokinase. Then again, glucose 6-phosphate, here we will use the different enzyme, that is the glucose 6-phosphatase. And then after this enzyme using, we will get formation of finally glucose from glucose 6-phosphate. And we have synthesized the glucose here from the pyruvate. So those three irreversible steps, they are there in the glycolysis. For that, we have used the different enzymes in the glycolysis, and we have synthesized the glucose from the pyruvate. Pyruvate is a non-carbohydrate compound. Now, in the definition only, we have covered that the gluconeogenesis is the synthesis of glucose from the non-carbohydrate compounds like pyruvate, lactate, glycerol, propionate. So from pyruvate, how glucose is produced, synthesized, we have covered. Now the second non-carbohydrate compound is lactate. So from lactate, what will happen? From lactate, first we will get the formation of pyruvate. Then pyruvate will enter into the mitochondria. In mitochondria, it will convert to oxaloacetate. Oxaloacetate then convert to malate. And again, same procedure will follow till we get the formation of glucose. So from lactate also, we get the formation of pyruvate. Now, one more important point here is then next the non-carbohydrate compound given is definition is the glycerol. So from glycerol also we get the formation of glucose, like from glycerol we get formation of dihydroxyacetone phosphate, then glyceraldehyde 3-phosphate, then fructose 1,6-biphosphate, and then we will get formation of glucose. So from glycerol also we can get the synthesis of glucose. From glucogenic amino acids also, see these are the glucogenic amino acids. From these amino acids also we get the some intermediate compounds, and then from that we get the malate. and then we form the phosphoryl pyruvate, and then we get the formation of glucose. So this is known as the gluconeogenesis, that is the synthesis of glucose from the non-carbohydrate compounds, that is pyruvate, lactate, propionate, glucogenic amino acids, glycerol, etc., is known as the gluconeogenesis. Here also I have mentioned the gluconeogenesis from the amino acids. So from these amino acids using we can synthesize the glucose. From glycerol, I have already told you, from glycerol we get first formation of glycerol 3-phosphate, then glyceraldehyde 3-phosphate will form, and then glucose we will get. Then from propionate, first we will get formation of succinyl coenzyme A. From succinyl coenzyme A, then we get the formation of malate, and then from malate we will get the glucose, like that. So all the steps will be same. Then from lactate, now this is the important point, gluconeogenesis from lactate. I have already told you this point, that from lactate we get first formation of pyruvate, and then the next step will be same as I have told you. So especially this formation of glucose from the skeletal muscle lactate is known as the Cori's cycle. Okay, so this is also important point. So when we get formation of glucose from the skeletal muscle lactate, that is known as the Cori's cycle. Lactate is produced by the active skeletal muscle, and it is a major precursor for the gluconeogenesis, because gluconeogenesis occurs in the liver. Okay, and the lactate is produced in the skeletal muscle, but in the skeletal muscle the enzymes, that is glucose 6 phosphatase, fructose 6 phosphatase, are not present in the muscles. So this lactate is transferred to the liver, and then in the liver we get the formation of glucose. Okay, so that's why the synthesis of glucose from the skeletal muscle lactate is known as the Cori's cycle. This is also the important point. In the skeletal muscle the lactate is produced, and then this lactate, as the enzymes are not present here for the gluconeogenesis in the muscles, they are absent, so lactate is transferred to the liver. In the liver gluconeogenesis will occur, glucose will form, and then again glucose is transferred to the skeletal muscle. This is known as the skeletal muscle Cori's cycle. These are the references, and this is known as the gluconeogenesis. That is the formation of glucose from the non-carbohydrate compounds.